Okay. Uh, Today our lecture will be about evolutionary biology. What can it teach us about finding life beyond Earth? Yes, our distinguished speaker tonight is David Baum. He's a professor of evolutionary biology at University of Wisconsin, Madison. About evolution, I think somebody said, maybe Alan Turing, biology is a science, evolution is a fact. So, I'm a physicist, I looked for evolution in a big image. We call it cosmic evolution. From hydrogen to human and beyond. From Big Bang up to now, up and more, which is the study of many developments and generation changes in assembly of and compositions of radiation, matter, life, intelligence, space, and time. For me, evolution is bigger than the biological evolution. Biological evolution is a special case. We are living on the earth. We studied the biological evolution of the earth, but in big image, we are living in the biological, cosmological evolution from Big Bang up to now. So I'm sure there's a life in some place in this universe. And according to Fermi paradox, because we are living in a huge universe, and distances is huge, and also time. Time, we are talking about 13.8 billion years from Big Bang up to now. So, and our life, our recent life, the 20th century, we are talking about hundreds of years with this technology or less. So this is the difficulties. We don't have a solid evidence of another life outside Earth. So I don't want to talk a lot about this topic because we have a specialist. Let me introduce Professor David Baum. He's a professor of evolutionary biology at the University of Wisconsin, Madison. Please welcome, sir.
Thank you. Thank you, really. I really appreciate the opportunity to talk to you all. For me, it's very exciting to not only talk to scholars in different disciplines, a lot of you are in the more physical sciences, but also to talk to scholars from all around the world like this. This is a real pleasure and honor. So, as you heard, I'm an evolutionary biologist. So my background is very squarely in the biological sciences. So I have had a career of more than 30 years studying evolution on Earth, looking at living organisms around us, particularly plants. I have a tradition of studying mainly plant systems. But over the last 10 years, it became clear to me that evolutionary biology has a fascinating connection to the biggest of questions, which is how do we have life? And so what I decided to do today was not give you a very long talk, not give you a very technical talk, but try and explain, really answer this question. What does evolutionary biology tell us about life and about the origin of life and about the distribution of life in the universe? I hope to leave plenty of time for discussion. I would love to pick up some of the themes that just came up about whether we should be seeing evolutionary biology as part of a broader notion of evolution that includes cosmic evolution, and how we should maybe think about the origins of technology, which I'm not going to touch on in my explicit talk. But to get us started, I thought it might be good just to pull up a definition of astrobiology. So this is the definition of astrobiology provided by NASA. And it defines astrobiology as the study of the origin, evolution, and distribution of life in the universe. So if you read that literally, it's basically saying that there are three co-equal components to the study of astrobiology: origin, distribution, and evolution. I'm going to take a view that actually this structure is incorrect, that actually evolution encompasses both of the other two features of astrobiology. That both to understand the origin and to understand the distribution of life in the universe is to understand evolution. So let's just start by thinking about this idea of distribution. So the question of where life is in the universe requires that we ask the question: if we find something somewhere on some other world, within our solar system or if we have sophisticated methods, or we have interstellar travel in exoplanets, we find something, the question is always going to be, is it life? Does it provide evidence of life? So what is that question? So I'm going to propose that the right way to think about a question, whether we have evidence of life, is to ask, is what I am seeing simple enough that it could have arisen spontaneously in its current form? Or is it so complicated that in order to have its current form, it has to have accumulated change and complexity over a long period of time over evolutionary history? So just to give you an example of the thinking, on the screen, there are two kinds of objects. So the one in the middle is a pumice. Of course, a pumice is, we know it's a porous rock. We know how it can be formed. It has a structure that, but the more we study it, the more we understand that this could arise spontaneously on a volcanically active world like our own. What about these things on the right? Well, these are sponges. Sponges, as you know, are the remains of living organisms, marine sponges. And at first glance, we might think that they were similar, but if we studied the formation of those structures, we would quickly find that they are integrated into a living organism that is tremendously complicated, that grows in a way that pumice does not grow. And we would conclude that this sponge, however simple it might be in the scale of life that we perceive around us, this sponge could not have just emerged spontaneously on the Earth without a long history of complexification. So the question of where life is in the universe is intimately connected with how we define and understand life. And I would say that that question is also a question that asks us to understand, what is it about evolution that allows complexity to accumulate? And how can we recognize the products of evolutionary history? So that's distribution. What about origin? Well, really the rest of today, I'm going to be talking about origin. So what the challenge in explaining the origin of life is that when we look at the organisms around us today, all of them are too complicated to have emerged spontaneously in their current form. So we obviously, if we look at something like a bird or a human or a tree, it's obvious to us that these have so many sophisticated and complicated mechanisms at the molecular and the structural level that we cannot conceive of a natural process whereby they could spring into existence in their current form. Or maybe less obvious, and was not actually obvious until the late 19th century, is that the simplest forms of life, such as bacteria, are also tremendously complicated. When we dive into the workings of even the simplest life, we find very complicated molecular mechanisms. In every cell, we find very specific molecular components performing very specific functions for the well-being of the cell. There are particular lipids that form membranes. We have the genetic material, the DNA, which is read into a transfer material, messenger RNA, and then that is converted into proteins. And the proteins work together in complicated ways to allow the whole cell to function. So when we look at a modern cell, it is a tremendously complicated thing. It has been estimated that the simplest cell, if you just count up the number of distinct components in that cell, it's similar in number to the number of distinct nuts and bolts and wires and pieces that make up a very sophisticated technological object like a 747 airplane. So the simplest cell is a similar order of complexity to a plane. And as the astronomer Fred Hoyle pointed out, we know from first principles that it is impossible to imagine a tornado blowing through a junkyard and blowing together a fully functional 747 plane ready to fly. We know this can't happen. And for a similar reason, we know that a genetic cell, like a bacterium, could not possibly arise in one spontaneous leap of improbability. It required some long history of complexification to get even the simplest cell. So the way that we have to think about the origin of life is that there had to have been some simpler but evolvable system that it originated spontaneously. So it had to originate spontaneously because by definition, it can't have evolved, because if it evolves, it's already an evolver. So the first evolver had to be something that arose spontaneously, had the capacity to complexify over time, and eventually give rise to the complex life that we see around us, like genetic cells. And so the question of the origin of life is a question of what is a first evolver? What does it look like? How does it work? And how could it complexify to manifest the features that we see in today's life? Features such as having genetic systems, having cellular organization. So to contextualize this, let's just put a timeline on the history of life on Earth as we now understand it. Now, for a long time, it was thought that there was a long gap between the emergence of the planet Earth as a water-rich world and the appearance of life. And this is because the main way that we could detect life was by finding fossils in dated rocks. And until not long ago, a couple of decades ago, the earliest evidence of life were macroscopic fossils that dated in the order of about 700 million years ago, 0.7 billion years ago. Whereas the estimates are that the Earth formed about 4.6 billion years ago, with liquid water being present on the Earth by about 4.2 billion years ago. So under this timeline, there is a long window during which life could have originated. If there's a long window, then there is more opportunity for an improbable event to occur at least once. So the view that there was a long waiting time for the appearance of first life supported views that treated the origin of life as a very improbable event that might have required very specific circumstances for the appearance of the first evolver. However, over the last couple of decades, geological sciences have made huge strides in closing this gap. So there is now strong indirect evidence of life much, much earlier than previously thought. So the problem that geologists face is that the longer the oldest rocks, in order to have record of life, a rock needs to be a sedimentary rock. It needs to be generated by weathering and erosion, generating sediment that can then be compressed into a rock. But that rock has to be formed and it has to persist without being melted again or subducted deep into the earth. And there are very few rocks that are sufficiently old and sufficiently able to retain a record available on the Earth. And in those rocks, though, when they are found, many of these early sedimentary rocks do have structures that have been interpreted now as being evidence of microbial life. So here is an example on the screen of a microfossil dated at about 3.5 billion years ago. It's obviously it's distorted because it's been, the rocks are so old, they've experienced heating and other disturbances. But there is now a good body of evidence conclusively showing that rocks as old as 3.5, 3.7 billion years old have evidence of microbial life. Now, earlier than that, there are no sedimentary rocks. However, there are ancient minerals like zircons, which can entrap organic material. And there can be clues in the material they trap. So this image, the second image I have here, is of a zircon dated as about 4.1 billion years ago. This zircon is formed in a volcanic setting, but it can have inclusions that trap as a material, including carbon. And analysis of the isotopic ratios in this carbon have led geologists to conclude that it is biogenic. It has passed through life. Life tends to enrich for light carbon. So through these sources of evidence now, the general consensus is that life originated very early and very quickly. In fact, at this point in time, there is no evidence of a period when life was absent on Earth. It could have taken 200-300 million years for the first living systems to appear. For all we know, it could have taken a week. We have no way to know exactly how long it took for the first life to emerge, but it was apparently quicker than was thought for a long period of time. So this change in understanding in geology has had an impact on the way that origin of life researchers and evolutionary biologists have had to think about the origin of life. If life evolved relatively rapidly, then it must have been, and it must have been simple, then we start wondering if perhaps life does not require very particular and rare events to get started. And it supports the possibility that actually life is easy and spontaneous and inevitable, even in parts of the universe that resemble Earth, in having some of the essential components. For example, being chemically out of equilibrium thanks to solar flux from the sun and radioactive decay in our planet's core. With the right, at least on Earth, we know we have certain key ingredients like water and carbon. And so there is been a shift, I think, in the astrobiology community into believing that simple life, not technological life, not multicellular life, not intelligent life, but simple life of some kind may be easy and abundant in the universe. And so that is the perspective that I think has emerged. And at the end of this, I'd be interested to know what you all think about this perspective. So taking that as a starting point, we then have to ask the question, okay, so we now believe that something simple arose that was capable of evolving. What was it? And how could it complexify to give rise to modern life and its mode of evolution, which is called Darwinian evolution? So how did something simple arise, complexify over time, and eventually give rise to the units that undergo Darwinian evolution? And this way of thinking is premised on the idea that something capable of Darwinian evolution is too complicated to have arisen spontaneously in a short period of time. So in order to understand this, it turns out we need to do some rethinking about what life means, what is life, or what are living systems in their simplest form, and what is needed for evolution. And so I'm going to take a little bit of time to kind of walk through some ways that I and many people are coming to rethink life and rethink evolution to help us understand this transition from geochemistry to biochemistry. So if you ask somebody to describe life, most of the time, people will describe a living organism or a living cell. It is a human bias, I think, to divide the world up into discrete units and to embody those and to see those units as that which we're trying to explain. So it's very tempting to equate, as I already did, a bacterial cell with another object like a plane. And because cells do something, they appear to have agency, they appear to have purpose. We often equate them with machines like cars or airplanes. And that is a very convenient way to think about our life as it exists today. But when it comes to thinking about how life originates, this way of approaching it has some problems. Now, there are commonalities between a cell and a plane in that both of them have to do work. Both of them have to conform to the laws of physics, and to do work, they have to use free energy, generate entropy. And in both cases, actually, we can say that there are some similarities. What they do is they use a chemical disequilibrium in the environment. So, for example, a cell might use sugar, which is an oxygen, which it takes in from the environment. These are chemically out of equilibrium. It can then control a reaction between them in such generating waste that is less out of equilibrium, thereby generating free energy that the cell can use. Similarly, the plane does the same thing using jet fuel and oxygen to generate actually the same waste. So they each do work they do work using energy from the environment. But what's important is the work they do is fundamentally different. A plane's work is whatever function we built it to do. In this case, carry passengers through the sky. A cell's work is really no more, no less than making more cell material. So what a cell is doing is making more protoplasm, protoplasm being the matter inside a cell. And so what a cell is fundamentally, if you want to think of it as a machine, is the machine for making more cell. So whereas a plane does its work, it moves around, a cell uses that energy disequilibrium in the environment to make more cell. And that new cellular protoplasm, it doesn't have to in principle, but it often does split to make two cells. But notice here that the protoplasm is continuous. The protoplasm, the matter that makes up a cell, has all come from the prior cell. And this is a fundamental difference between cells and machines, is that all cells come from other cells. All protoplasm comes from other protoplasm. Protoplasm, living systems of chemicals that take used energy disequilibrium in the environment to make more of those chemical mixtures. Those chemical mixtures are continuous in time. Every cell comes from another cell, whether it be bacterial cells dividing like on the left, or we have an egg and sperm, two living cells coming together to make a zygote. In all cases, life is continuous. And this was famously put in Latin by Louis Pasteur, omne vivum ex vivo, all life from life. So this requires that we actually slightly rethink what life is. Life is not a set of objects. It is a continuously growing protoplasm. A way, this is cartoony, but a way you can think about it is that back 4 billion years ago or so, somewhere, somehow, a first evolver emerged. It was a protoplasm. This protoplasm grew, used energy in the environment to expand. We can imagine it almost like it was a gigantic amoeba that grew and covered the planet over time. That gigantic protoplasm is still alive today, and it's basically been immortal. It's not, there's been no, sure, certainly pieces of it have died, but the protoplasm as a whole has been immortal. And it's immortal because it feeds on an energy disequilibrium that is constantly being replenished by the sun. And so what we have then, when we look at life, what we should be looking at is not objects, but a system that is planet-wide and immortal, that just keeps growing and expanding and growing and expanding. And while it's growing and expanding, it's also breaking up into smaller pieces. And those smaller pieces, being evolvable in their own right, can end up following different trajectories and ending up in different looking pieces of matter. But it's still one living protoplasm today. So when we look at all of the diversity on life around us, what we should really see is not just a set of beautiful, wonderful, amazing organisms doing different things. We should also see the commonality of all of it. It is all the same protoplasm connected through time back to the origin of life. And I personally find that to be inspiring, amazing, but also essential to appreciate if we're going to explain the origin of life, not as the origin of an object, but the origin of a system, a planetary system. It has been said, and I love this quote by a few people, that the life is not something that evolves on a planet. It is something that happens to a planet. A planet actually becomes a living system. That protoplasm is integrated and exchanging matter and information effectively with the parts of the planet that we consider non-living. And there are many instances of this where the planet's behavior fundamentally changes as a result of life. We know about the production of oxygen in the atmosphere. We know it's less well known, but living systems change plate tectonics. Living matter can lubricate tectonics and help tectonics. And there are many other ways that there is feedbacks between this protoplasm and the rest of the planet, the so-called non-living parts of the planet. So Earth is a living world. Okay, so to understand this, how all life in these different forms can be traced back to one living protoplasm, I thought I should just very briefly, and at a very shallow level, just clarify what evolution is, how it works. So, and this is necessary because what I want to do is show you briefly how we can rethink evolution and understand evolution such that it could have occurred with much simpler ingredients than a cell. But if we have cells, then those cells grow and divide, and they grow and divide to make populations. So we have a population of cells, and they all are connected through common ancestry. They all trace back to the same protoplasm, but it's now in different packets. And over time, rare events happen. A rare chemical reaction can happen in one. So here we have a population of cells. And let's suppose that a rare reaction happens, and one of them, there is a change in the chemical makeup of the genome. So that genome of that particular bacterium is a different molecule than the other ones. So instead of being, it's chemically different chemical. That chemical difference has an effect on the cell. And perhaps that chemical difference makes that cell slightly better at functioning in the environment in which it occurs right now. If that is the case, then that cell might be more efficient at growing and dividing. And so its descendants might ultimately take over the population, and the blue ancestral form might be lost. That is called natural selection, Darwinian evolution. And we know from, more than 150 years of research that this fundamental mechanism can explain all of the diversity we see around us today. Once we add in the fact that the populations can periodically split into independently evolving lineages. And so given a sufficient amount of time, this basic mechanism can allow for the incredible, explain the incredible diversity of living organisms we have around us today. From mammals, and various animals. This particular graph is a little biased towards animals. It implies that somehow they're special and the purpose of evolution, but they're not. All living organisms today are equally evolutionarily advanced, including bacteria, archaea, plants, fungi, and so on. And so, this graph, I should say, is showing time running outwards from the center. So combining this process of population splitting and evolving independently with this process of adaptation and rare mutations, where mutations can either increase or decrease complexity, but whether they are enriched or not is independent of that. A more complex form will often be favored. As a result of that, complexity has been able to accumulate for 4 billion years or so to give rise to everything we know around us today. And that is not really controversial. What is controversial is, how did you get the first thing capable of Darwinian evolution? Because as you may have seen, in order to be a Darwinian evolver, there are 2 things you seem to need. You seem to need some kind of cellular organization. You need to be some structure, some object that grows and divides and grows and divides and creates a population. You also need to have some mechanism like a genome where a rare reaction, a mutation, can change it, and that change can be passed on to offspring. And that change has the potential to affect the ability of the cell to grow and divide and grow and divide. So in order to understand the origin of life, then, the tricky thing is explaining how you can get adaptive evolution without cells and without genes, and how that kind of non-Darwinian evolution could give rise to cells and could give rise to genes. And that is basically what my research program addresses. How could simple chemistry begin evolving adaptively and give rise to cells and genes? So I think we have made a lot of progress in understanding these 2 things. So starting with genetics, and I'm going to give you a very, I'm not going to give you a very in-depth answer at this point. At the end, I'll trust that if you have questions, you'll get into them, and I have extra slides I can go through. But I want to give you the feel right now of the principle. Historically, biologists and biochemists have viewed genes as being very special, as being uniquely able to carry information between parents and offspring. What's special about a gene life cycle is that if we have a particular, let's say, DNA sequence, that DNA sequence can be copied into a complementary sequence. that complementary sequence can then separate and be copied to generate a second copy of the original sequence. So once a gene sequence is generated, the expectation is that barring another rare chemical reaction and another rare mutation, offspring will get the same sequence as the parent, which means that once that gene is activated, it will tend, all things being equal, to persist into future generations. That has been the viewpoint in biochemistry, is that that is special. That is a special feature of genes that is not true of regular chemistry. But it has now become clear through a lot of theoretical work in chemistry that actually that principle is found broadly in any autocatalytic chemical cycle. An autocatalytic cycle is just a cycle that feeds back on itself, so that once activated, more of its components are made, more of its members are made. So looking on the left half of the screen, we see a little hypothetical autocatalytic chemical reaction. In this reaction, this chemical Z or Z reacts with food to make a different chemical X. X reacts with food, undergoes a branching reaction to make one Z and one Y. That Y then reacts with some different kind of food maybe to make another Z. So what this means is at the minute we have an environment with one Z in it, we get 1X, then 2Z, then 4Z, then 8Z. We get an exponential increase in the number of Z's and X's, provided we're in an environment that has plenty of food. Now, the interesting thing about this reaction cycle is that even if you had an infinite amount of food in the environment, nothing would happen until a rare reaction generated a Z, an X, or a Y. And so in exactly the same way that a rare reaction is needed to generate exactly this sequence from some other sequence. So it turns out that the ability to carry information from one generation to the next, to carry information through time, is not unique to gene. It's a general property of autocatalytic cycles that require a rare feeding event to be initiated. And it turns out some work from in my lab and others have shown that if you look at the kinds of chemical reactions that would have occurred on the early Earth before there was life, there are uncountably many autocatalytic cycles. They often look a lot more complicated than the simple one I've shown. So here is one that we've sort of documented. In this reaction, there are reactions shown in with a little R at the beginning. There are chemicals, and then the chemicals are colored based on whether they are food, shown in blue, or waste, shown in gray. And in this particular treatment, we've included visible light and infrared light as though they were chemicals. We can do that in these analyses. So the upshot is that what we think happens pretty spontaneously and pretty easily in a spatially structured world is that rare reactions trigger new autocatalytic chemical systems. So, and to make this sort of visually easy, I'm imagining here a case in which we have a food-rich ocean and a series of mineral patches. And these mineral patches absorb certain chemicals, and there are certain reactions that can happen on the surface due to catalysis. Now the imagining here is that a rare reaction seeded a particular autocatalytic chemical cycle shown in green on this surface and on this surface, and maybe a different one was seeded on this surface. Now over time, maybe the green one, there could be once you have one autocatalytic cycle activated, it can allow other ones to be activated that can feed on them. So now maybe a yellow one emerges on that one, maybe purple ones on here. And through a kind of process that resembles the way that ecological communities of organisms, of animals and plants and fungi and bacteria complexify over time, we would expect ecosystem patches in the environment to become colonized by increasingly complicated sets of autocatalytic systems to complexify spontaneously. And any of those mixtures of autocatalytic pieces that are better at persisting in the environment and not getting washed off the rock, or better at spreading from one patch in the environment to another patch in the environment, those would be favored. And so in much the same way that cells that have a gene that makes them fitter will tend to increase in frequency, carrying that gene to higher frequency. Similarly, these little ecosystem patches, the ones that have combinations of autocatalytic cycles that make them better at surviving and spreading, will also tend to increase in the environment. And so we can have a very simple primordial kind of adaptive evolution occurring before we have genes and before we have cells. So that gives you a sketch of the way we think about the origins of evolution before cells and genes. What about the origins of cells? Well, it turns out, I've sort of shown you this already, that you don't need a population of cells. A population can be generated by the environment, such as what I just showed you, a series of patches in the environment. It turns out that when you model this mathematically, you can show that as soon as you have spatial structure in an environment, you have the essential ingredients for selection. And so we've done in my lab a lot of modeling of chemical reaction systems as they might complexify in spatial structures, such as continuous arrays or these patchy arrays, like I showed you a minute ago. And so that mechanism explains how evolution can happen. It also gives us a hint as to how cells might have originated. If we have a spatially structured environment where different ecosystems, different chemical ecosystems, and I'm using the word ecosystem here to refer to a localized place where there are many autocatalytic systems interacting with one another, similar to the way that many species in biology will interact with one another in a forest or in a grassland or something, or in a coral reef. So we have a lot of these ecosystems, and selection's favoring the ones that are good at persisting and the ones that are good at getting around to available new sites. One very good way of getting around is to make a propagule. A propagule here just means a packet of some kind that carries with you members of all of the essential autocatalytic cycles that are needed for that ecosystem to function. And so those propagules are a way to get from one patch to another, and those propagules can later evolve and become regular cells. So that is how we think that you can understand the origin of life as a gradual process of adaptive evolution in the absence of cells or genetics. At some point, cellular encapsulation evolved, and we think it could have been through selection for dispersal ability. And at some time, genetic systems originated and eventually gave rise to the kinds of living systems we're familiar with today. So I want to check the time. I should probably, so just to kind of put this in the context of what I do, and I'm not going to go into detail on this, but I just want to show you that by breaking the origin of life problem down into a number of steps, all of which relate to evolution before genetic cells, we now have a way to actually study it in the lab systematically. So in my lab, we study this full spectrum of trends using both experiments and theory. So, for example, we've done a lot of theoretical work, collaborating with computer scientists and physicists and mathematicians to understand how chemical reaction networks can behave ecologically in an evolutionary way in the absence of genes, in the absence of cells. We also do experiments in my lab where we actually study out of equilibrium chemical mixtures to look for evidence of ecological and evolutionary dynamics, such as the kinds that we've been predicting. I already showed you, we do computer simulations to study dispersal dynamics, and we're using them to try and understand exactly the circumstances that would favor the origins of cell-like propagules. We're doing a little bit of work on studying heritability, the ability to evolve in genetic cells that don't have genes. So we can make them in the lab and we can study their evolution. And then we're also studying the evolution of genetics. First of all, trying to understand how it is that simple chemistry can give rise to the molecules that underlie genetic systems today, proteins and nucleic acids. And then we're using computer simulations to understand how those polymers that have catalytic functions can become enriched and organized into a genetic system. And then finally, we're now just about to start. We just got a grant to do some research to really directly study the origins of genetic systems in vitro, using microfluidic systems to actually do selection on cell-like structures that we can then give polymers to and see if we can get evidence of the emergence of genetics from scratch. So putting this all together, I'm going to say that, if we know enough about how life originates, and by how life originates, I mean, how do we go from simple chemistry to systems capable of Darwinian evolution, then I think we would know what aspects of life as we see it today were necessary and would be expected of any instance of life anywhere in the universe, and which aspects of life as we see it today are just accidents, things that happen to occur on this planet, but needn't have occurred in general. So when we look at a cell, which of these features would we expect to find elsewhere in the universe? Would we always expect to find nucleic acids? Or would we always expect to find amino acids and peptides and proteins? Would we always expect to find lipid membranes? And we can also then, if we understand the theory well enough, ask questions about whether we even need water. Are there other ways to be alive that don't require water? To be alive, to evolve complexity, that don't require water. So I believe, kind of going right back to the original question, that studying the origin of life is actually very central to looking and guiding us as we look for life elsewhere in the universe. So whether we look within our solar system, on Mars, on icy planets like Enceladus, or the amazing Titan, this is a rendering of the Dragonfly mission to Titan, which I think is supposed to be scheduled for 2036, which is a very exciting one for me. We may also consider the possibility that there are actually other living systems on this planet that we haven't yet found because we don't know how to recognize them, maybe deriving from independent origins of life that happen not to yield familiar signatures like nucleic acids. It's not directly relevant to the topic today, but I would just mention that we also think that if we can understand how chemical systems can begin evolving and complexifying in the absence of life, we might imagine a kind of chemical engineering of the future, which is fundamentally different than the chemical engineering we do today, in that it resembles breeding, like plant and animal breeding, more than it resembles traditional engineering approaches. So I guess the last thing I'd say is that even if neither of these things are true, even if my particular research doesn't shed light on life in the universe, at the very least, I'm excited to believe that we are learning something fundamental about how life got going on this planet, And to me, at least, that is a very fulfilling question to answer because, you know, life is just so extraordinary. So I just want to end by, you know, thanking my lab, my many collaborators, and I've listed a few of the main ones there, and my funders. So I've been funded primarily by National Science Foundation in the US, the NASA, the National, I should have forgotten what it stands for, Space Agency, National Aeronautical and Space Agency, and the Alpha P. Sloan Foundation, and by my own university, the University of Wisconsin-Madison. So I guess I'll leave it there, and if there's time for questions or comments, I'd be delighted.
Oh, thank you. Thank you so much, David, for the nice lecture and very simple and give us an overview about the origin of life in the Earth. And I hope that we can find beyond the Earth. Not yet sure, no solid evidence for life outside Earth up to now. Now, I think, yes, it's a time for Q&A. You can use the chat if you want to ask a question. You can write your question on the chat, or you can use the electronic hand, and we can give you opportunity to talk. Now we have Ahmed Abu Khadir. Please, Ahmed, welcome with your question.
Hello. First off, thank you so much for this presentation. The topic of life in the universe is a really interesting one. But I wanted to ask, I found that there are about 20 bodies in the solar system which contain massive amounts of water under either under surface or in their interior. That's a massive sample from just one planetary system. Do you think that other solar systems, in this case, would be star systems, would they have similar amounts of water in their planetary bodies, or is it just some kind of like cosmic fluke that our solar system has this much water? And can this water can water evolve? Can life evolve underground, or does the planet need to have some ocean on the surface for it to evolve? Thank you.
Thanks for the question. I can't really answer the first question, although my interactions, there's people in the room who probably can, but my interactions with my astronomer colleagues here lead me to understand that it's unlikely that our star system is unique or special in that regard, in having water. But the question you asked about whether you require the surface ocean of liquid water in order for life to emerge, I certainly don't think there's any reason to believe that. I mean, you certainly do need some kind of disequilibrium. You need to have some mechanism that is generating out of equilibrium feedstocks that can drive productive chemistry.
So like geothermal energy.
Well, thermal energy is a little less easy because it's, you know, it tends to, but, you know, tidal mechanisms, there's certainly radioactivity and sunlight. So even if the sunlight is weak and indirect, if it's perpetual, it can be generating disequilibria that potentially could allow the emergence of the kind of chemistry that would be needed for life. So the bottom line is we don't really know. Of course, we will not know until somebody, you know, finds direct evidence of living systems in some of these watery worlds. But my own intuition is that it's likely. It doesn't mean it's going to be easy to recognize, because I didn't say this, but I think there is no a priori expectation that we would find that independently evolved originated life would have DNA, making it a little bit tricky to know how you would recognize life on another world, especially because these probes don't, as far as I know, NASA has never sent a microscope into space. So even if they found cells, they wouldn't really have an easy way to see them. So the bottom line is, yeah, go ahead. Sorry.
They could detect its biosignatures, some chemicals it releases that easily decompose in the atmosphere. Oxygen is definitely possible, but I think there are some There could be some unknown geological mechanisms that could produce large amounts of oxygen. We just have to investigate further.
Yeah, you're quite right that, biosignatures are a big topic of discussion, and it's a little up in the air which, if any, would be definitive.
Thank you.
Thank you. Good question.
Thank you, Ahmed. One more question. Yes. Usam, please.
Yes, yeah, good afternoon. I'd like to ask a question. I'd like to know if there's been any research that has tried to correlate the changes in these autocatalytic cycles with the seasonal changes on Earth, things like changes in length of night and day, and the seasonal changes, and whether they could have affected those autocatalytic cycles we're speaking about.
I suppose the one area where there's been work along those lines is thinking about the origin of polymers and the potential importance of wet-dry cycling as a way to foster the emergence of polymers. The particular chemistry of the polymers that life uses, like peptides and nucleic acids, involves every time you make it longer, you release water. And so drying organic molecules has been shown to promote the production of, polymers. And so some people have speculated that a lot of the early steps of polymerization occurred, for example, in pools where the cycling of rainfall, day-night cycles, and the like were very important for driving the appearance of long polymers. And so when we do experiments, I mentioned some experiments we do on looking for autocatalytic polymers. We use wet-dry cycling, similar to what might have occurred in a natural setting. I don't know if that answers your question, or is there something more general that I'm missing?
Okay, thank you. Marwan, please.
Hello, assalamu alaikum, everyone. Professor David, thank you. Actually, I couldn't be here from the beginning, but I have a question, if you don't mind. So, as per I understood, you said life is not something that evolved on a planet, but it is something more that something happens to the planet. This is a quite and strange. I'm not sure how much strange that is, but this leads to a question, and also I need also a request, if you will. But let me first just ask: does this mean life cannot happen to something else? A planet is a must to happen. And the other thing is, I would like to see one of those simulations, computer simulations that you developed in your lab, if any available right now. Thank you very much.
Your first question is a very interesting question. Is it? And I don't know entirely how to think about it, but there's an issue of scale. Yeah. So the chemistry that we're really focused on tends to be very local. Autocatalytic chemistry requires that molecules interact, and so they're always going to be local. I guess the reason that I think we view it perhaps as a planetary phenomenon is autocatalysis by its very nature tends to grow to fill the available space. And if the available space is a planet, it will expect it to impact the whole planet. And so there is a sense in which the chemistry of the whole planet changes at that level of observation. I don't think it's somehow saying, you need a physical space. You can't occur in the abstract. So does it have to be a planet? Clearly not. It can be by planet, I guess I should say a world. It could be a moon. So yeah, So I guess I'm not sure if I've really answered that. What kind of simulations were you specifically interested in? More on the early steps of like evolution in unstructured systems, or more like spatial structure?
Yeah.
Let me
Actually,
go.
because this actually takes me to those ages in the 90s, when I was using the basic language to do those evolutions, so the trees and those things. So this is why I'm very interested in seeing something new related to the evolution and as computer simulations. Yeah, please.
Yeah, I mean, in terms of the simulations per se that we have, I'll put a couple of names in. So for the very in the chat, I'll put a couple of names. If they will they go to everybody, or will they go to? So so if you search, so if you want to know about sort of the very beginning steps, sort of the ecology and evolution of chemical reactions and spatial structures, collaborators of mine, Emily Dolson, if you just look for these names, you will find a few papers. So sorry, my typing isn't always so fast. And then I actually gave a citation to it. We have a actually, I think it now has a third author.
Emily Dolson, yeah.
Yeah, and then if you look at this, we have a on the spatial structure, we have an archive preprint up at the moment. It's Kempes. I'm sorry to spell that wrong. So those are some of the examples in terms of the computer simulation of genetic systems, which I'm quite excited about. Those we haven't published yet. I could show you some preliminary results if you're interested. But I could just, things like this, where we can sort of study how genetic systems can sort of bootstrap themselves into existence. But it's probably not time to go into it in any detail.
Thank you.
Welcome.
Yeah,
Thanks for the
thank
question.
you. More questions and Q&A? Looks that everything is okay. Maybe last question about what's... Okay, Hannah, yes, welcome, please.
Good evening, Doctor. I'm sorry that I wasn't able to attend the lecture. I had another engagement, so I missed it out, unfortunately. Anyhow, I had a question, maybe not directly related to your lecture. It concerns the difference between chemosynthesis and photosynthesis. See, when we are talking about photosynthetic life on Earth, we need, of course, sunlight, enough sunlight to do this job. Now, the question is, since we found life at the depth of oceans that are away from sunlight, which utilize chemosynthesis. Now, can't we find the same thing, let's say, in the oceans of, let's say, some moons, Jovian moons, for example, like Europa and other moons? And therefore, this idea of the Goldilocks zone, why are we always seeking the Goldilocks zone or the habitable zone? Why don't we search for conditions similar to the oceans, to the deep depths of the oceans here on Earth? That's my question.
I mean, I totally agree. I mean, in terms of the history of life on Earth, I think there's a very vibrant debate as to whether the earliest living systems, the earliest sets of cooperating autocatalytic cycles, would have been photosynthetic, chemosynthetic, or heterotrophic, meaning they would just feed on the organic matter that was being made spontaneously by sunlight and lightning and other sources of energy. And I don't want to, there's no reason to get into that, but I totally agree that all you need is a sustained chemical disequilibrium. And there's some very good reasons to think that the kind of disequilibria that are found deep in the oceans, that are ultimately driven by fission radioactivity in the Earth's core, those are likely to be occurring elsewhere and those could easily drive the, provide the out of equilibrium chemical mixture that some kind of nascent life needed. So yes, I agree with you that we shouldn't just assume that you need to have an exposed liquid ocean with lots of sunlight.
Okay, thank you so much.
Thank you.
Thank you. More questions? Yep. Me, I have a question. The last question, it's what's your personal speculation about life outside Earth? Personal speculation about life outside Earth. I'm not talking about science. Your speculations.
Sure. Yeah. That's good. Okay, so I'll just give a quick anecdote that I was at a NASA workshop on biosignatures in, what was it, like March or May? No, I think it was May, sorry, earlier in the year. And at the end of the workshop, somebody said, okay, everybody raise your hands if you think there is life elsewhere in our solar system. And everybody put their hands up. So I guess I am in that view of believing that microbial life, simple life, things that have accumulated some evolutionary complexity are probably very abundant in the universe. Now, until recently, I would have said that the likelihood of complex life, like particularly technological life, was perhaps irrelevantly low. But I'm beginning to wonder. I think that our Earth is very special in that it has been sort of basically stable for 4 billion years, which is a long time and has allowed for quite a lot of innovations to occur. If there are other worlds that have been this long-lived, I can imagine that the probability of them hitting upon a series of probably long and unusual steps, hitting upon organisms of some high level of complexity, it may actually be quite high. It just would depend on how long they persist in sort of the same sort of evolving state. So bottom line is I am very open to thinking that life is abundant in the universe, and intelligent life, obviously much, much, much rarer, but it may still be present elsewhere.
Thank you so much. Yeah, now I think no more questions. Right now is the time for, let me thank first David Baum. He's professor of evolutionary biology at University of Wisconsin, Madison. Thank you so much for the nice presentation and very fruitful discussions. Now, I think it's the time to have a photo together, if you don't mind. But if you stop sharing to have the full screen for our people, please open your, switch on your cameras, please. Please switch off your camera. Marwan, would you please help us to have a photo for
Yes,
everybody?
of course, yes.
Yes, please.
So الجميع لطفا نفتح الكاميرا
Yeah,
إكراما
please,
بهذه الجلسة
everybody switch
الجميلة.
on your camera
Yes.
here.
اللي
Smile. Yeah, everybody.
اللي بيرغبوا
Yeah.
يكونوا معنا. جميع الكائنات الحية تكون معنا الآن.
Yep. Yes. Yeah,
Yes.
switch on.
Okay.
Open your camera, please. Sally, String Theory, Thomas, Maria, Sabah, Adli.
Everyone. Okay.
Everyone. Yeah.
شوية تعديلات بسيطة.
Okay.
Last moment.
Najah. Rimah. Everybody open your camera. Yes, please.
Okay.
Okay.
We need
Yeah.
last
Smile.
Okay. Just a few words, Professor David. So we get the green frame, the speaker frame,
speaker frame.
Okay.
on your picture as
Okay.
a presenter. Yes.
Got it. I will. There you go. You have it.
Taken. Thank you.
Okay. Thank you so much for everybody. And thank you so much, David, for the nice and very fruitful presentation. And see you again.
Yeah. And if
I
anybody
have been in
has,
Wisconsin, by the way. I have been in Wisconsin several times.
oh, good. Well,
Milwaukee.
next time, come say hi. And if anybody has any
Okay.
questions, feel free to email me or
Okay,
comment.
thank you so much. Thank you so
Okay,
much.
goodbye.
Bye-bye. Thank you, everybody. Thank you. Bye-bye. Thank you. Have a nice time. Bye-bye.
Thank you.
Thank you.